Okay, so I always like to start any lecture out. We've talked about a bunch of stuff before. Any questions that anyone has about anything we've talked about? Right, we've kind of given you introductions. Jacqueline very nicely set me up with some basic background in quantitative genetics. I'm going to talk about, in 2 lectures, trying to find something about the underlying architecture. But before I do that, do people have any questions going over their extensive notes or thinking about things about anyone that's given a lecture before? Again, I encourage you to do that, right? Take notes during class and don't be bashful. Likewise, catch us at lunch. Mike's around through next Wednesday. I'm around basically the whole time. So, feel free to catch us. So what I want to do is talk about detecting QTLs or quantitative trait loci. And the upshot is I'm going to tell you why this has been largely a failure. Setting you up for the next lecture to tell you what we can do about it a little bit differently. Okay, there we are. Oops. So here's kind of the motivation. A lot of the questions we're going to be facing when we talk about polygenic response has to do with the genetic architecture. If 2 traits have the same heritability, you can predict the response over a couple of generations. But as you go longer and longer and longer generations, one trait may basically end up plateauing out in a small population. The other trait may keep on going. Why? If one trait has a very large number of genes, a small effect underneath it, and the second trait has a modest number of genes of larger effect, everything else being equal and population size large enough drift isn't an issue, you'll get continued response out of the population with the more underlying variation. Why? Because the sites aren't being fixed as selection proceeds. So architecture is really, really critical. That's the joint distribution of allelic effects and their frequencies, and also number of loci. That's how we'll define architecture. And the revelation is we can actually, there is a delay here. We can actually get a handle on this now by using molecular markers to tag or put a flag post on regions of the genome. And what QTL mapping does is it puts a flag in a big chunk. Like the big chunk would be, southeastern India, that it's somewhere in there. GWAS allows you to make that flag cover a much, much smaller areas, as we'll talk about. So the thing is, we can use what markers do to get some information what the underlying causal sites they tag do, but there's some subtleties there. And we do this in 2 different ways. Historically, we've used linkage mapping, and that's what's often called QTL mapping. That'll be the subject of most of today's lecture. We also use association mapping, also called GWAS for genome-wise association studies. I'll introduce a little bit of this, and we'll flesh that out tomorrow. But the key here is for linkage mapping, I need to follow the ancestors of particular individuals. I need to follow their, and usually it's one generation like a family. We can also do it in a pedigree. I'm using linkage information, as we'll talk about. The regions that are tagged are often on the order of megabases. So you put a flag, and what you said is that in this region, which is maybe 20 to 40 megabases, that's a lot of genes. In this region I flagged with a marker, there are 1 or more factors that influence variation on the trait you're considering. And one of the limitations here is these are usually based upon line crosses. You can do pedigrees, but usually line crosses. And the early studies here came out of agricultural systems. You take a low-performing maize and a high-performing maize, inbred lines, you cross them, you make a fairly big family, and you score that family. So the limitation on QTL mapping is how big is your pedigree or how big is your family size. And for most things, you're lucky if you get a family size of 300. A really high-powered experiment might have 2,000 to 3,000. That's great for certain things, but it's not good for other things like fine mapping. We'll talk about that. The other subtlety thing here is that when we score variation in QTL mapping, because this is generally, not often, but not always, but generally, crossing 2 inbred lines, every initially segregating allele has frequency 1/2. That really changes the architecture, because instead of having this L-shaped architecture that Michael talked about a lot of alleles have very low frequencies, you now have every segregating allele has frequency 1/2. And what that does, for example, is it creates the possibility of more dominance and more non-additive variation. If you have the same underlying genetic model mapping genotypes to phenotypes, and you have equal allele frequencies, then you tend to load more of the variation onto non-additive components than you do in a situation where the architecture is, where the genotype's the same, but now you have rare alleles. Why? You're fitting a best linear fit. You only have a couple of points. You fit everything on the additive. So the net result is that because you're looking at crosses between population and fixed differences, you're actually scoring a different type of variation than when you're looking at within population variation. And so comparing the results from between population crosses and within population variation, there's subtleties involved because a lot of this between population differences may have been rare alleles of large effects that are fixed by selection that you wouldn't see, you'd see them in a segregating, but they wouldn't account for much variation. So we're going to talk about linkage mapping. We'll also talk about association mapping. It uses something called linkage disequilibrium. We'll talk about that. And the key here is now I can take a random population sample. So the bottom line is anytime you're trying to do gene mapping, if you have the option of spending more money to get more markers or more individuals, where should you spend your money? Markers, individuals. You've got a big bunch of money, you've got a big grant. Where should you spend that money? Individuals, for a couple of reasons. There's a practical cheat reason. If you score individuals, you can always keep DNA and go back and get markers when they're cheaper. But the bottom line is that unless you have recombination between those markers, it doesn't matter how dense that marker map is. So you always want to get individuals for power to get recombination, breaking things up. And when you go to a GWAS, now instead of looking at a single sample from a pedigree, which is limited, you're now basically unlimited. The largest meta-analysis is about 7.5 million individuals looking at height. You can do that because in a GWAS setting, you're not using relatives. We'll talk about why that comes up. So we're going to talk about linkage mapping and association mapping here. So background reading, I think I posted these, but it was kind of chaotic because it wasn't clear until like 12 hours before I left if I would be coming here or not. So if I haven't posted these, let me know. And by the way, Jacqueline said it was Lynch, Vischer, and Walsh. I won the arm wrestling contest with Michael, so I got my name first on here. I think he was worried about all the mistakes I was making, so want to have me up front. But chapter 5 talks about linkage and linkage disequilibrium. Chapter 18, we'll post these if they're not posted already, talks about QTL mapping. And chapter 20 talks about GWAS. So I'm going to give you kind of a sketch. There's like 60 pages here and 60 pages there of all the gory details with worked examples and all the references. So I want to kind of give you a feel for what's going on. And if you want to really delve down into specific things, you can ask questions or you can look at some of that material. Sure. So this is the 2000, this is, so yeah, it's, there's, yeah, so basically there's a lot of weird terminology out there. So I'll use the Peter Vischer terminology. So Peter Vischer called Lynch and Walsh 1998 as the Old Testament. And the 2018 book, so the 1998 book deals with basic structures in quantitative genetics. The 2018 cockroach killer, because it's great for killing cockroaches, talks about selection. We call that volume 2. Yeah, but volume 1 here, this is the revision of our 1998 book, which is going to be even bigger. So this basically, this is the first 21 chapters of the first half of that. And there's about 1,500 post-2000 references in there. So this, by the way, should be out by the end of the year, pending getting comments back quickly enough from someone by Oxford and dealing with Oxford. But this will be out soon. And anyhow, you have a draft copy of some of those chapters. And if you don't, send me an email, I'll make sure they're on there. That's the background. Here's the overview. We're talking about genetic markers, SNPs, STRs, and whole genome sequencing, because we're kind of moving to here. I want to talk about linkage and linkage disequilibrium. Boy, they sound like they have linkage, but they're actually rather different. And we'll talk about the consequences. Then I'll talk about linkage mapping. We'll talk about marker trait association, also called QTL mapping, marker trait association. We'll talk about hypothesis testing. We'll give some examples of some limitations. Then I'll talk about something which is really important but often misunderstood, called the Beavis effect, after a famous paper by Bill Beavis. It's also called the winner's curse. That term, by the way, comes out of bidding for oil leases. So it comes out of the economic literature, and it's been captured by the epidemiological literature, winner's curse. Then I'll finish up by talking about association mapping, marker trait association, correcting for population structure. And I believe we have a practical, the last lecture of today, that's going to go over how to use some software for that in more detail. So I'll kind of set that up a little bit. So part one, markers. Let's go back to basics. You can't get much more basic than this. This is what genetics was like until like the mid-1960s, arguably the mid-1980s. We had morphological markers. I had a classmate in graduate school that his hobby was seeing how many homeotic mutants, these are development-changing mutants, he could put on one single strand of Drosophila and have them live. So he had some really bizarre, but you could put 3 or 4 on. So when we only had these visible markers, and of course here are the 7 genes that Mendel used, and they were all nice because you had a dominant and recessive. So wrinkled was double recessive, green was double recessive, round could be a homozygote or heterozygote, but this was what people had throughout a large part of quantitative genetics, was basically morphological markers, which was terrible. You can't do much with them. Now there's an exception. Sachs in 1923 crossed a heavy spotted and a light unspotted, heavy meaning seed weight, bean lines, and he noticed in the F2s that while not all heavy beans were spotted and not all spotted beans were heavy, if you were heavy, you also tended to be spotted. And those were 2 morphological markers. Morphological number 1, the spotting locus. Marker number 2 was the gene that was involved in determining one of many seed weight. So there's an association created by linkage. So we need to find markers in order to look for those associations. So the original markers are here, but then we got sophisticated. In the 1960s, we had electrophoretic markers based upon how different proteins runs on gels with specific stains. That was the introduction to molecular population genetics. Then it got replaced. Now we're kind of in this framework here. We usually look at SNPs, single nucleotide polymorphisms, STRs, simple tandem arrays, and now of course we do whole genome sequencing, and you often pick these features out. There are other features like duplicates, etc. But just to remind people, here is a SNP. There's your favorite DNA helix. And the SNP means in a sample of chromosomes in the population, some have a G here, a GC pair, some have an AT pair. There's a polymorphism in that nucleotide, in that single nucleotide. You also can have STRs. I stole this picture from a crime website. See who's guilty. So when people talk about a DNA test at a crime scene or for paternity, it's almost always using these STRs, these simple tandem repeats or short tandem repeats. And now what you're looking at is these are scored by, this DNA sequence now is scored by how many copies? 7 copies here, 4, 12. So we call this a 7, a 4, and a 12 allele. At the second one, 6, 19, 14. So now what we're scoring is a size variation, and those fall in discrete classes. And so STRs were used fairly early on, and they're now being replaced by SNPs. Let's talk about some of those reasons. So let's look at STRs. Let's look at the pros first. If I have 13 highly polymorphic STRs, I can distinguish any 2 individuals on the planet that aren't identical twins. This is what the CODIS marker data system uses in the US for DNA profiling, and that's because at an STR, I may have 10 or more alleles, and those 10 or more alleles are all at roughly equal frequency. So I pick a random site, there's a 90% chance it's a heterozygote. Heterozygotes give you information. These are great for linkage studies within an extended family, but the problem is they have a high mutation rate. So let's imagine that I have a new mutation that arises here. It's this lovely water, and now it's on a STR that's a 6. Well, if I have a low mutation rate, the only way that association is broken up is by recombination. So if this is a SNP, that SNP ain't going to mutate. It's only going to be broken up, this association, by recombination. If I have an STR, this 6 could mutate into a 7 or a 5 or a 6. Mutation rates are about 1 in 250, 1 in 1,000. So people like STRs, also called SSR, simple sequence repeats, they're good over very short time scales, but over longer time scales, they're unstable in the sense that if you use them for tagging a tightly linked region, that association is broken not by recombination, but rather by mutation. Now, conversely, with SNPs, the cons is they're less polymorphic. You typically only have 2 alleles. In theory, you could have up to 4. Usually, the chance of getting a heterozygote is modest, maybe 15 to 20% if you choose your markers right. But the advantage is they have very low mutation rates, and they're very stable. So these are great for looking at long-term associations. Like if you're doing an association mapping, use SNPs. If you're doing linkage mapping, you can use either one. People often have panels of STRs around from previous work, in which case they're useful. These are great for looking at population structure. They're not good for doing fine mapping over many generations. Now, the reason that SNPs are now becoming feasible is it's now trivial to score hundreds or millions of them on a single chip pretty cheaply, a couple of bucks, if you have the right system. If you don't have the right system, wait about 6 months, and the DNA sequencing technology is getting cheaper and cheaper and cheaper. I think a whole genome sequence for human now is like under $100 bucks. It's a good quality sequence. And the read lengths now are telomere to telomere, not these little fragments you put together. So these are the type of markers people typically talk about. There are others like transposable element insertions. When we talk about tagging a site, usually have one of these type of markers. If I'm looking at linkage mapping, I can use either one. If I'm looking at association mapping, I want to use SNPs. Why? Because I'm looking at long-term associations. Associations generated hundreds of generations in the past. SNPs retain those. STRs have those mutate away. So, basic genetics. Let's go to linkage. Linkage is what makes genetics interesting. If we didn't have linkage, all this fun stuff we do with modern genomics would be problematic because we're not typically following a causal site. We're following a site that co-segregates with that causal site, and that's linkage. So Mendel had 7 traits. He has 7 chromosomes. Now, to be fair, I think 2 of the traits are on different arms of the same chromosome, but the odds basically of picking 7 traits in Ps with their chromosome number and have them all be on different chromosomes or different segregation groups is about 1%. So Mendel was either divinely inspired, because he made beer after all, or he probably threw out some of his data. So what happens with independent assortment? Well, here's a simple idea. Yellow and round versus green and wrinkled. And what you have here is in this parent, you get 4 types of gametes. You get a single allele from each locus, so big Y, big R, et cetera. And then over here from the other parent, and under independent assortment, so the probability of big Y, big R, little r, they're independent. It's just this joint probability. You can do a Punnett square and figure these things out really quickly. So Mendel's second law was independent assortment. However, if you have dependent assortment, you now have to do conditional probability. So in particular, if I wanted to compute a little y that is yellow, big R that is homozygous round, that means I've got to get a little y, big R from one parent, little y, big R from the other parent. So when you have linkage, instead of following alleles around, you follow around gametes. You can think about gametes as chromosomes, or more generally, I have a sperm cell with haploid sets in there, what's in there. And so with independent assortment, life is easy. You just look at each locus independently. But then Bateson and Punnett came along, I think this was 1906, and they were looking at flower color and pollen shape. I think this was P. And the pollen shape was, it was either like a rice grain, kind of elongator, or kind of spot-like. And what they did was they crossed a pure breeding, that means completely homozygous, round, a purple long with a pure breeding red round. And here is the expectation in the F2 from the 9 to 3 to 3 to 1 Mendelian ratios under independent assortment. Here's what they saw. If you look at this, you see it's an excess of big P, big L, and little p, little l. You do not get independent assortment. This is the first paper that said we've got this potential for things called linkage. And here's the basic idea. So in the heterozygote, F1, you had a parent from the big P, big L line, and a chromosome from the little p, little l line. And if you don't get any crossing over, you get either one parental or the other parental gamete. If you do get crossing over, you get a recombinant gamete. The closer these are, the higher chance of recombination, of not having a recombination going. So it's basically just what I said there, the basic Punnett data. And so what would happen under independent assortment? Under independent assortment, each of these 4 gametes has frequency 1/4. But if you have linkage, you have something called the recombination frequency. And the recombination frequency says, what's the frequency of the 2 classes that are recombinant? Well, that's C. Since they're equally frequent, each is C/2. So if you can look at the gametes from a single parent, you can right away tell what the linkage phase was big P with big L or was big P with little l. Because right away what you see is you get 2 classes that are both above 1/4. That gives you the linkage phase, and here big P, big L on one chromosome, little p, little l on the other. And then the smaller fractions, both less than 1/4, the sum of that is the recombination frequency. So basics of linkage, most of you have probably seen it before. Went over a little bit quick, but just to make sure we all have the same kind of terminology background. So linkage is following the gametes from a single parent. That's what you're following, because if you have linkage, then you should see alleles that co-segregate from that parent. These don't show independent assortment. Linkage disequilibrium, often more correctly called gametic phase disequilibrium, concerns a population sample. You can have linkage without LD and you can have LD without linkage, although when you have this case, it usually decays away fairly quickly. So let's talk a little bit about that. Suppose in this room, I sample all of chromosome 1. So I've got 60 samples of chromosome 1. And on that, I look at what's the frequency of the marker A in that sample, what's the frequency of the marker B in that sample. Then I ask, what's the frequency of chromosomes that contain an A and a B? If this relationship holds, A and B in your population sample are independent. What that means, for example, is big A tends to be with big B as often as big A tends to be with little b, adjusted for allele frequencies. So if I don't have, that's called linkage equilibrium. If I don't have that, I have linkage disequilibrium. And it's simply the frequency of that, I say gamma, you can think about chromosome, that makes it easier for you. The frequency of that chromosome type minus its linkage equilibrium value. So it's a sample from a population. There are lots of forces that can generate these non-random associations. We'll talk about some of those. And so this D here, if D is positive, it means that this gamete is overrepresented, and over time it's going to decline down to its value here. If this is negative, this gamete is underrepresented, and it will increase in time. So one of the complications that arises when you move from single locus population genetics to multi-locus population genetics is linkage disequilibrium. You can't simply follow single loci one at a time. You have to follow what their gametes do. Why? Because gametes' frequencies can change without any other forces other than recombination acting on them. Allele frequencies are stable under Hardy-Weinberg, but under Hardy-Weinberg single locus, gamete frequencies can change until you get this relationship satisfied. Question? Yes? You can have LD, yes.
Because of sampling, is that?
So great question. So let's go old school. I think I'm gonna be the first person to use the chalkboard. Sorry about that, Nick. But by the way, for Nick, you'll notice that they've got boundaries on here just for you. You can't go below there or below there. Suppose I cross this. Suppose I cross AB times, and let's make them on different chromosomes. Little a, little b, little a, little b. So in the F1, what do I get? I get A, B, and I get A, B on here. But in here, this gamete only has big A, big B. This gamete only has little a, little b. So in this F1 population, I've got LD, even though they're unlinked. Now that goes away in the next generation. And so one of the ways you get LD between unlinked sites is with population structure. You can also get it with selection. It generally tends to be small, and it's removed very quickly, as we'll talk about in a second. But how many of you have used a program like Structure for population? So if you use Structure basically works by looking at little tiny LD signals across a large number of loci, and then tries to structure populations so those LD signals go away, because those LD signals are generated by mixing two populations. So one way you can get LD between unlinked sites is when you've got things like population structure. And again, you can generate it that way, it gets removed extremely quickly. So let's look at the difference between LD and linkage. So let's look at, here's my sample of parents. So what fraction is big A? Well, I've got 4 out of 8 copies are big A, so big A is a half. What about big B? Same thing. So the frequency of big A is a half, the frequency of big B is a half. Under linkage equilibrium, frequency of big A, big B should be a quarter or 2 out of 8, and I do see that. There's 1, there's 2. So I have linkage equilibrium in these parents. That is, there's no non-random linkage phase. But now let's look at parent 1. I have linkage. I get an excess of big A, big B. Parent 2, I have linkage, excess of big A, little b. Parent 3, excess of big A, big B. Parent 4, excess of big A, little b. If I look at any parent by itself, I see this pattern, that's linkage. However, if I pool all these gametes together, that signal goes away. Why? Because the parents are in linkage equilibrium. And so even though the gametes from any one particular cross is going to show an excess, when you average over all the linkage phases which are there, those cancel, that signal entirely goes away. Conversely, now I've got 3 big A, big B. That's a departure. I expect 2 out of 8. I now have 3 out of 8. Same thing. Now when I pull the gametes, I get an excess of big A, big B, little a, little b. Why? I have an excess of big A, big B, little a, little b parents. So linkage is what do I see in the gametes from a particular parent. LD is what do I see when I sample the population. Now when we talk about linkage mapping, you're going to basically make crosses and look in those crosses. When I talk about a GWAS or association mapping, I'm going to sample a population. And the reason it works is if I have markers which are close enough, that is a kilobase or 10 kilobase, I can get long-term historical associations that will persist over thousands of generations. And we'll talk about that in a little bit. So linkage and linkage disequilibrium both have this term C involved. The reason they have that term C involved is this is how any initial disequilibrium decays. It decays by 1 minus C. So if linkage is tight, it persists for a long amount of time. If linkage is loose, that is near a half, it decays away extremely quickly. So the connection is their rate of decay depends upon the recombination rate if it's not being regenerated by some other force. But other than that, linkage disequilibrium is a property of a population that I've sampled. Linkage is if I look at the gametes from one specific parent. So we can exploit linkage if I can follow the gametes from a parent like in a cross or in a pedigree. Linkage disequilibrium means if I have LD there, because markers are close enough together, I can then exploit it in a very large population. So association mapping, as we'll talk about a bit later on, exploits linkage disequilibrium. So those are important concepts, and they're very easily confused because they both involve the term linkage and they involve associations. One way to think about it, and if this confuses you, just ignore it, right, is you can think about that pool of gametes as being your reference population. In that case, you would have LD in that pool of gametes. There'd be non-random associations. We typically, though, talk about LD as a population or a sample property, and linkage is the gametes from one specific parent. So now let's use that concept of linkage to map QTLs. Any questions so far? I should warn you. Yes, please. So the great question. So the question was STRs are highly polymorphic. Could you expound upon that? So what happens is when you have these, think of these as little railroad boxcars. I've got 6 boxcars up here. I've got 5 boxcars up there. During DNA replication, they can easily misalign, right? Because they're stable. When they misalign, polymerase reads through them and generates copy number differences. So because of that, they have very high error rates. Whereas SNPs have all sorts of controlling factors on them. So a SNP was 10 to the minus 9, 10 to the minus 11 is what SNP rates are, ballpark. SNPs don't mutate, I mean, for all intents and purposes. STRs will mutate like crazy, especially if they're good for a couple of generations, but the mutation rate is very high. Because their mutation rate is high, if I have an association of my fist and a marker 4, if that's a SNP, that only gets broken up by recombination. If that's an STR, it can be broken up by mutation. So when we're looking at long-term, you always want to use SNPs. And people are moving to SNPs now because they're much cheaper to score. It used to be STRs were cheaper to score, but now SNPs are cheaper to score.
Does that mean then that SNPs are more population-specific than STRs, which tend to be less population-specific?
Yeah, this is a great question. Are SNPs more? So the answer is kind of yes, because you don't use FST when you have SNPs. You use something called GST because you can back mutate. So people still do population structure with STRs. The difference is that they use fewer of them, so they kind of average out, where with SNPs, as we'll talk about, you could get a big hit on a SNP, a single SNP, because it's population specific. And that could really mess up your GWAS. We'll talk a lot about how we adjust for that. So great questions, keep them coming. Sure, so we're talking about linkage. Again, great question. There are no bad questions. And by the way, it's important you ask questions, because if you don't ask questions, I assume this is all old hat and review, and I can speed up. And I'm acting on like 1/6 speed right now, so you don't need to speed up unless you really need it here. So we have this key parameter called the recombination frequency. And I think this was your question. The recombination frequency simply says, if I look at the gametes from a parent, what fraction of them are recombinant? So going back to our example here, I look over all my chromosomes from here. What fraction involve these events? That fraction, let's say it's 12%. It means of all the gametes I look at, only 12% involve one of these two types. That's the recombination frequency. And so as the distance between the two markers gets larger, everything else being equal, the recombination frequency gets close to 1/2. As the distance gets smaller, the recombination frequency gets down to 0. Was that your question? This slide here?
No.
Okay, further up. I mean, down in this slide deck. Up.
There were circles and
Oh, you mean here, excessive parents? So here, this one right here, you mean? No?
there is a place you're showing some circles and arrows downwards where you have gametes A, B, A, B.
I'm not following. We'll go back to this. We'll go back to one.
I think it's further.
Oh, down the slide. Okay. Yeah, up and down. So larger, higher in number. Here or the next slide? This slide here?
The previous one.
This one here. Okay. I didn't talk much about that, but so this D is the disequilibrium between A and B, and it simply says you look at the frequency of big A, big B chromosomes, and this is what you expect if A and B were independent. So this is the linkage equilibrium value. The observed value minus the LE value is your disequilibrium.
No, my question was, there is a time you're using excess of A, excess of in the next slide after this.
Next slide.
Yes.
Going to slide 19.
Yes.
Okay, this down here you mean? So these show linkage. So big A, big B, and little b tend to co-segregate. So this parent has a big A, big B, little b linkage phase. They show an excess of parental gametes. This has a different linkage phase. There's a different excess. So if I look at this family by itself, I see big A and big B co-segregating. Look at this family, big A and little b, big A and big B, big A and little b. Because big A and big B are in coupling, big A and big B are in coupling. Yeah, this is like, so maybe I should, this notation means one chromosome has big A, big B / the other chromosome has little a, little b. So unless you get a recombination, that chromosome stays intact. So that means mostly you get big A, big B, little a, little b. Here you get big A, little b, little a, big B. Yeah. Okay. No problem. Some good ask questions. Not a problem. Okay, let's pull this together. So that thing we called C, the recombination frequency, we actually typically don't use that. We use something called centimorgans. They're very similar when C is small, has to do with mapping functions. The chapters talk about that. So we talk about 20 to 40 centimorgans. It means a recombination frequency of at least 0.2. And as a rough, crude estimate, and there's huge variation, you can crudely think about that for a lot of organisms, 1 centimorgan equals a megabase. That varies within you and it varies between species, and Mike will go on forever about it's a bad approximation. Just gives you a handle to think about that. So QTL mapping tries to detect small chromosome segments. Think about a chromosome segment basically being about 100 centimorgans. So a small segment would be a piece of that, and that could be still a big chunk of the chromosome. It could be 20 to 40 megabases or more. So this is a relatively crude level of resolution, but nonetheless, it basically says, it plants the flag. This chromosome arm here has 1 or more factors that influence trait variation because they're segregating in that. And these are performed either using inbred crosses or sets of individuals, and they follow from this idea about an excess of parental gametes. I mentioned Sachs, 1923, the first person that did this. We knew how to do QTL mapping. Actually, Payne, 1917 was the first paper, chromosome arms. We knew how to do QTL mapping, a century ago. The reason we haven't done it until recently is we didn't have the markers, right? To do QTL mapping, you need to have multiple markers on each chromosome, and you want those markers not to affect the morphology of the traits you're scoring. So if you're looking at height markers using P, well, you can't use tall and short because that's going to mess up your other markers, right? So molecular markers have the nice feature in that you can get a lot of them, and they don't impact, ideally, the traits we're looking at. So what we can then do is look for what we call a marker trait association. So I basically do, here's the idea. Here are 2 inbred lines, and the line represents a chromosome. So M, big M, and little m are 2 marker alleles that I score. Big Q and little q are alleles that influence my trait, but I don't score those directly. So if in the F1 I get a big M, big Q, little q, then in the F2 I get these gametes here. If M and Q are unlinked, then all 4 of these gametes have frequency 1/4, and big M doesn't give any predictive value if you have a big Q or a little q. But if I have linkage, let's imagine I have a recombination rate of 2%, then big M has big Q, so I see a big M, big Q almost half the time, and I almost never see a big Q on a little m. So what this would basically say is that 98% of all chromosomes that carry the marker big M also carry big Q. How would I use that operationally? I take my first marker, I break it up into homozygote 1, homozygote 2, heterozygote, and down here is homozygote 1, up there is heterozygote, over here are the other homozygote. I have 3 classes. I simply do an ANOVA, or do a regression on number of markers and ask, is there a difference between the phenotypes if I break individuals based upon classes for that first marker? Big M, big M, big M, little m, little m, little m. No. Go on to the second marker. No. Third marker, no. So I scan across the genome until finally I get a marker where, hey, big M are much larger than little m, little m. That then plants a flag and says somewhere around the big M marker are 1 or more factors that are influencing the trait of interest. And I can tag that marker that way. That's what QTL mapping basically does. It creates a marker trait association. So the logic is actually really straightforward. When you have linkage, if you have a causal locus next to your marker, they tend to co-segregate. So the marker genotypes then predicts that you have if I have a big M versus a little m, if there's linkage, big M is going to have on average, a different QTL genotype at the linked marker than the little m will. And that's the fundamental idea behind QTL mapping. If you get that, all the other stuff is just kind of details. So it's actually very straightforward. It's a natural extension of linkage mapping, but now instead of scoring two traits, green and yellow, what I score is I score the first marker, then I put individuals into marker categories and ask if the traits differ between those marker categories, a marker trait association. The exact same thing is done with GWAS. So a really simple idea. We mean geneticists, knew how to do it early on, but we didn't have the markers to do it. It wasn't until the 70s and mainly the 80s people started thinking about actually doing this with markers. So we have some mathematicians here, so I thought I'd throw a little math in there for you. When I'm doing QTL mapping, I'm trying to do two different things, detection and estimation. When I'm trying to estimate, what do I want? Because just flagging, hey, around this site, there's one or more QTLs. Well, estimation would involve, if there's a single causative site, what's the recombination frequency C from your marker? Also, if I've got 3 potential QTL genotypes, which I would have when I cross 2 inbred lines, what are the mean differences? So detection says somewhere in this chromosome region is 1 or more sites. Estimation says what's their position and what's their effect. We get at that this way, and they're more complicated models. They all come down to this basic feature. This is your old friend conditional probability. The probability of seeing, and I've written this with all sorts of scrub strips to make this generalizable. Don't worry about that. Probability of seeing a particular QTL genotype given the marker is the joint probability divided by the probability of the marker. Now you're going, well, wait a minute now. How can I use this? I don't know that. The way you do these is you suppose that Q is at some recombination distance C from the marker and ask what the probabilities are. Then you just simply run over all those probabilities, all those C values, and find a maximal value is how we kind of do this. So in the F2, here are the frequency of those 4 genotypes. There's linkage. And let's imagine I have a big M, big M. So if I have a big M, what's the probability that I'm big Q? Well, it turns out 1 minus C squared, where C is the unknown distance between the marker and the QTL. Big Q, little q is 2C, 1 minus C, little q is C squared. What if C equals a half? So I get a 1 to 2 to 1, so I get no information. If C is near 0, it means every time I have a big M, I have a big Q. So basically this recombination frequency C says how much of the signal from that causative site maps onto the marker itself. So we would use these as something like this. So what's the mean value for a marker? Well, suppose the QTL genotype is that. There's the conditional probability given the marker. That's where the recombination comes in. This is the mean for that marker. So we can then estimate those. So a simple case, if I have a single QTL linked to the marker, and that QTL has the difference between the 2 homozygotes of 2A, then the M1 marker homozygote, sorry, MM minus little m, that difference divided by 2 is A, half the distance between the 2 QTLs, times 1 minus 2C. C equals a half, there's no effect. If C equals 0, you're estimating basically this difference estimates 2A. So if I get some value here, and that value is small, it could be because they have tight linkage, C is near 0, and A is small, or it could be that A is big, but C is near a half, so that number is small. So this is the simplest scenario. I've got a single QTL linked to my marker. More complicated scenarios follow simply by bookkeeping from this, and chapter 18 gives you all the kind of bookkeeping details. So let me say one more thing and ask if there are questions. So what would be the statistical model for that? This basic model is really easy. It's a one-way ANOVA. So I have my 3 genotypes, marker genotypes. We'll call them 1, 2, and 3, big M, little m, little m. I simply fit this and ask, are these B's all 0, or is one different from 0? Is there a marker effect? So the simplest way you can do it. It's a simple model. And the nice thing about this is it gives you detection. If one or more of the B's are significant, it says that marker plants a flag on a chromosome segment where there's 1 or more segregating QTLs nearby. The tricky part is how do we go from that to estimation? Well, I gave you one simple way to do it in this simple scenario where I could then take this difference and then try to estimate A and C. And if I know, if I've got additional markers, I may be able to get a bound on C. But we typically don't use a single marker to do this. You can use pairs of markers, but more generally use something called maximum likelihood. And I'll save that in a second. By the way, you can extend this. How about epistasis? This is the effect for marker I, effect for marker K. Is there an interactive effect here? So for example, if this term is significant, then you have a linkage to A. If that term is significant, you have linkage to B. If this is, then you have an interaction between them. One of the interesting things is there are cases, these are called main effects. There are cases where the main effects are small and the epistatic effects are large. So strategies for searching for those are covered in chapter 18. So do people get the basic idea, at least about detection, that you can flag a region using a marker? So you have a marker trait association. Trait values differ over different marker genotypes. Now, again, this isn't over most marker genotypes, only over a small fraction that are near a QTL. So most of these won't show a difference. And that's where power comes in. They actually may show a difference, but your sample size is too small to pick it up. That's one of the limitations you have in QTL mapping. So questions about the basic strategy? Again, it just follows some linkage mapping. There's kind of some bookkeeping in terms of how you figure out stuff. So most of this is done with maximum likelihood. How many people have heard of maximum likelihood? Who invented maximum likelihood? When in doubt, say Fisher, right? If you ever stuck in statistics, just say Fisher. And if you're wrong, at least sound sophisticated, you know. So if you look at the history of statistics in genetics and genomics, early on we had something called method of moments. And that is, I'm trying to estimate a variance and this thing of, you know, xi minus x bar squared divided by 1 over n minus 1. That's expected value equals the sample variance. Those were nice methods for estimation. They didn't require a lot of assumptions. The next wave is likelihood. And likelihood basically says, let's go to a normal distribution. Go over here. And when we have a normal, I admire Jacqueline can draw the bell-shaped distribution smoothly. So you've got a mean, you've got a spread sigma squared, you've got a value here. So normally we talk about the phi mu of sigma squared, it's the density function, right? And that's, as you all know, is 1 over square root 2 pi sigma squared, e to the minus x minus mu squared over 2 sigma squared. So normally when we think about probability is, I tell you what mu and sigma squared are, then you ask, well, what's the chance of getting an x of 6? In maximum likelihood, we turn that on its head. We say my data are that I see 6, 4, 2, 1. So I plug in 6, I plug in 4, I plug in 2 and 1. I get a product of these things. Now, once I have the data, this is now an unknown function of mu and sigma squared. So instead of treating the parameters as being fixed and trying to ask how likely am I to see a given observation, I take my observations as being fixed and say, given that observation, how likely are those unknown parameters? So the basic idea is really pretty simple. I then, when I do this, I have a two-dimensional surface, mu and sigma squared, and I search that surface numerically, maybe. Then I find the highest value. Highest value is also called the maximum, and that would be called the maximum likelihood estimation, right? Then I look at the curvature around that. Why? Suppose it falls off very sharply. Is my estimate very precise or very imprecise? Well, if I move a little bit, it drops off. It's very precise. As the surface gets flat, I've got a lot of uncertainty. So the high point is the estimate. The curvature, and you can take curvature in multiple dimensions. You can have high precision one dimension, low precision the other. That curvature gives you an estimate of the sampling error. And suppose I think my model has mu of 0, sigma squared 1. Here's the height if I plug mu of 0, sigma squared 1 with my data. Here's my MLE. The difference in those two is a likelihood ratio, and I can do hypothesis testing. So the nice thing about likelihood is it takes the three goals that people have historically had in statistics, estimation, precision of estimates, and hypothesis testing, and puts them into a single framework, and all you have to do is tell me what the density functions are. And so that's a model assumption, but you get more out of the data. The good news is a lot of things that you're used to dealing with are actually likelihood estimates. Things like the Gauss-Markov theorem for linear regressions, that whole sort of thing. So we can use this in QTL mapping. Oh, I should mention here. So when I do something like this, that assumes the trait is continuous. But a lot of interesting data are discrete. You're dead or alive, you have the disease or don't have the disease, so-called dichotomous characters. We can handle that by either, for example, making a presence-absence table here and simply doing a simple chi-square, or you could use something called the Cochrane-Armitage trend test, which basically looks if there's an increase in here and uses fewer degrees of freedom. People use both. So the Cochrane-Armitage test basically asks, is there a constant amount for each copy of big M? And again, chapter 18 talks about that. So you may see people do that. You also may see people use logistic regression. If your data is binary, people often use logistic regression. We'll talk about that a little bit in GWAS. So if you look up a paper, continuous data, people typically just don't say what they do. With discrete 0-1 data, they may use a chi-square test or an Armitage test or logistic regression. So likelihood, I kind of mentioned that. What's the likelihood function look like for QTL mapping? So this is now your new friend, right? This is a normal distribution. So if I have a value of z and that's the mean and that's the variance, that's the height of that curve. So what I have here is, let's imagine I've got 3 QTL genotypes, big Q, big Q, big Q, little q, little q, little q. So I've got 3 different values there. I'm going to assume they have a common variance. This term here, the chance of that QTL genotype given the marker, we've seen that before. This is where the location comes in. That's where C comes in. This is where the mean differences come in. So now I have 1 parameter, 2 parameters, 3, 4, 5 parameters. With I plug in all my data, so I basically then have a likelihood that's now a function of 5 parameters. The 3 means, the common variance, and the C value that comes out over here. And then I simply solve that. People often use something called the EM method to solve it by quick iteration. And I can solve for that. I can get the likelihood estimate of the position and the effects. It's called the mixture model. Kind of what I said before. So this is kind of the nuts and bolts of just see how it's done. Position information, recombination fraction comes in here. If I've got several QTLs, then the genetic map comes in there. So position information comes as conditional probability, effect size comes in here. And I typically plot that, let me skip ahead here. I typically plot it like this. This is called the profile plot. If you pick up a typical paper on QTL mapping, you'll see something like this. This is 3 traits. And what you see here is trait 1, we'll get this threshold line in a second. Trait 1 has kind of no effect. Trait 2 has no effect. Trait 3 has a big peak there. So the high point of that peak, right there, that's the MLE for the QTL position. You can look at how quickly it falls off and get a support interval. And again, there are different ways to do that. Chapter 18 talks about that. And so you might think, well, there's got to be 2 QTLs here. You see this little peak here? Well, you have to be careful. Once you fit this first QTL, put that as a cofactor, rerun the model, these peaks often disappear. So this is a profile plot, and this is what you would typically see. And the significance threshold you can get by permutation testing. So my last lecture at the end of next week, I'll be talking about things like permutation testing, combining p-values, meta-analysis, just kind of some general tools for high-dimensional data that you'll get when you deal with genomics problems. So that's the basic structure you see. And what you're doing here is this threshold basically says that you get a value C that's significantly different from a half. And so you have evidence for 1 or more QTLs. Now the problem is it's often more, and we'll talk about some of the complications that can arise from that. So when you open up a paper on QTL mapping, you see that. What they're basically plotting is this likelihood ratio. Why? Because that the full likelihood stays constant, so you basically just plot it across there. So this is a projection, basically, of map position. Once you get the map position, then you can go back in and plug in what the effect sizes are. So most QTL mapping does this, and if you open up a paper on QTL mapping, you'll see a bunch of these figures. So questions about that? Again, I'm trying to give you a feel. I've skimmed over a little bit of the details. Mike always gets on my case about, Bruce, you're making the damn book too big. So I'll put the details in there and you can look them up rather than kind of grind them through for you here. So what you want to think about when you see a new approach is, do you understand the basics? The basics are, if I have a marker that's nearby a QTL, that gives me a signal, gives me a tag. I then should see the trait value differ as I go over the genotype classes for that marker. So I've detected it somewhere in that region. How can I then extract out more information? Likelihood gives you a way to extract out more information and map it a little bit finer. So questions or comments about that? Great question. So the support interval, so there's the typical thing you see is you take 10% of this highest value, that's called the one load support interval. Those turn out to be too narrow. You probably want about a 2 load. But the bottom line is, as you change your cross, you're going to change the likelihood shape. And all that matters is how far below do you go from that. So that one load support basically comes out of the fact that that corresponds to a chi-square value of 4.6, which is a 95% cutoff value for a chi-square 1. So it's a crude way of getting a 5% cutoff level, is how that's thought about. One of the nice things about this, let me talk about this significance threshold for just a second here. There's a great approach called permutation tests. Anybody know who came up with permutation tests? Fisher. So the application for gene mapping was Rebecca Dorsch at Cold Spring Harbor. Anyhow, I'll come back to Gary Churchill. And it's a really cool idea. And the really cool idea is, how do you get these thresholds for your particular setting, which has a certain set of markers and a certain cross, and your cross could be different from mine? The answer is super easy. Each individual, think of them as a vector. You've got the trait value up here, then you've got a vector of all the marker information. You want to keep that marker information together, because that tells about missing markers and about linkage, et cetera. So what you then do is you take the top of that vector, the trait value, and you just think of the deck of cards and you shuffle it over the marker scores. You've now randomized the trait value relative to the marker vector. Then you run your analysis and you find the highest value under that randomization where it's done in the neutral model. You do that several thousands or tens of thousands of times. Then you get a threshold. And what you say, for example, is only in about, out of 1,000, only, let's say 10 were above 2.2, and now you have your threshold. And you can do it chromosome specific, so it's a very nice way to go. It's not foolproof. You've got to know what you can randomize, and we'll talk about that on the last day. But that's how you can do this for very, very general crosses. A very nice approach because it takes all the idiosyncrasies of your data and naturally incorporates it. So permutation tests are a nice kind of tool to have in your toolbox. Now, when people do QTL mapping, you often see what's called CMI, or composite interval mapping. And the concern was, if you have multiple QTLs in a region, that tag can pick up a bunch of QTLs. So what CMI does is it basically says, here's the marker I'm testing between I and I+1. I've got markers out here, the blue boxes. If I put those markers out there in my model as cofactors, any QTLs out here or over there are automatically taken care of. So composite interval mapping gives you a way to basically make your QTL mapping more precise by basically putting bodyguards on your markers to make sure that if you get a signal here, it's only influenced by these nearby markers in the middle. Anything outside these markers is not touched at all. And the way you do that is simply add a little cofactor to your model. Again, details are in chapter 18. So when you see most QTL mapping now, it's composite interval mapping with maximum likelihood. And that's kind of what people have used since, you know, the probably mid-1990s. So we have this nice technology. You can see it's pretty applicable, right? All you need, are markers. That was a big deal when these were first done. It's now not a big deal. Then you need to make a cross and you need to be able to score your trait efficiently. Some traits are awful to score, like disease resistance, because it's so noisy. But if you have a trait you can score easily, you can do QTL mapping, and people have done this. When I grew up, they were just getting out of the what's called find 'em and grind 'em days of population genetics, where basically people at Lewontin and Hubby had discovered these molecular markers based upon protein variations. So you'd go out to your favorite organism, you'd get a whole bunch, grind it up, and run it. For some reason, they're often very tasty, like people did squid and clam and mussels. It's kind of a weird thing because you had to deal with the material somehow. And so that was the big phase, was do that. Then, 30 years later, QTL mapping came along. The big phase was to do QTL mapping on everything, because it was a nice, pretty straightforward approach to do. So what happened? Well, here's one of the early papers. This is Edwards et al. Out of NC State. And they looked at a whole bunch of vegetative traits in maize. And they had a bunch of what are called allozymes, these protein markers, pre-DNA markers, and a couple of STR markers. And what they showed, this is an average over all their traits. So for example, only about 2% of the markers accounted for 8 or more % of the variation. And once you get up over here, you lose power, right? Because you don't have any power to detect down this area over here. This probably goes up a lot more. And this kind of L-shaped curve kind of looks a little bit infinitesimal-like, and you have a lot of stuff that's pretty small, but you also have some stuff that seems to be kind of modest up here. This is what people would often call oligogenic, several genes, also semi-major genes. So when QTL mapping came about, this was not an uncommon pattern. And here's a classic example. This is Doug Chimke's work. So you have these two mimulus, Lewisii cardinalis, bumming bee and hummingbird pollinated. They're fully infertile in the lab. You can cross them by hand and get this. But in nature, you don't see them because they have different suites of pollinators. So a really, really cool system to ask about evolution of pollinators and the genes involved. And so there's the full bumblebee suite. There's the full hummingbird suite, everything in between. And they made these crosses and put them in the wild and all sorts of cool stuff. But their original QTL mapping found this. So Bradshaw and Chimsky. And look at over here. This is the fraction of phenotypic variation accounted for by the marker. Wow. Evolution is all by major genes, right? The smallest thing here is a quarter of all the variation was caused by a single marker. Petal carotenoids, kind of orange-red colors, 88%. Anthocyanins, kind of dark colors, 25 and 33. So if you look at this, these are all traits that were evolved in these differences in suites. And if you look at that, you go, my goodness. Obviously, infinitesimal model was dead. Those are major gene effects. Let's go out and do stuff with them. A lot of studies found these. Turns out a lot of them were artifacts. So in the mid-90s, the studies suggest that genes of major effect were pretty common. These are pretty common genes of major effects. These are over here. Let me go back up here. So the Edwards data, these are kind of major effects. 8% is a pretty big effect, but a lot of them were small, but other studies showed much larger effects. And so by the 90s, the idea was genes of major effect are pretty common. You're roughly L-shaped distribution of effects. You had many genes of small effects, often not detectable, and a few genes of large effects. And again, the caveat with these is this wasn't naturally segregating populations. These were populations that were forced typically by taking two highly divergent inbred lines and crossing them. So the nature of variation here is a bit different from what you'd see segregating in a population, but nonetheless, it suggested that changes are driven by genes of large effect. Well, what are some of the issues with that? To get that, we have to talk a little bit about power first. It's easy to detect a chromosome segment. It's hard to find map them. If I've got 300 F2 individuals, that's a pretty big family. It's remember, from a single cross, a QTL accounting for 5% of the variation. That's a big QTL. This is phenotypic variation. I can map that to a 40 centimorgan interval. That's about a chromosome arm. That's pretty good, but it's not fine mapping. If I want to map this same 5% effect to a 1 centimorgan, that's a megabase. That's still huge. I need 10,000 individuals. So QTL mapping isn't going to get you there for fine mapping. Also, the Beavis effect was discovered. And what Bill Beavis did was here's the simulation that did it. He had looking at maize, and had a simple model where he had 40 QTLs, each had an effect of about 1.6%. And then he ran the model a bunch of times, and then simply said, when I declare, that's the key point, when I declare a marker effect to be significant, how large is it? So if I average over all marker effects over all studies, I should get about 1.6%. But if I simply ascertain or bias the data by only choosing those markers which are significant, now what do you see? So look at how often that you saw markers that accounted for 15% of the total variation. That's half the time, where the actual effect was 1.6%. This is the Beavis effect. You're getting this because you have very low power, and I'll work that out in a second. When you have low power, when you declare a marker to be significant, you've almost certainly massively overestimated it. So a lot of these, for example, the Mimulus data were probably big overestimates. Another way to look at it, you can then look at power and how much you've overestimated it by. If power, let's say, is 40 to 50%, power simply means if I run the experiment over and over and over again, if the power is 30%, it means 30% of the time I'll declare that marker to be significant. And as you see, as your power gets low, so for example, here power is like 2%, my overestimation is 20-fold. And how do I know what my power is? I need to know the effect size. So you can't work backwards. Just because you have a big effect doesn't mean you've got high power. It could be an outlier. So where do these Beavis effects come from? Well, in epidemiology, this is called the winner's curse. GWAS has used that as well. I grew up in the Beavis and Butthead days, so I like the Beavis effect here. So here's the idea. Here is your true value, and this is a distribution of realizations for marker trait effects if you run that program again and again and again and again and again. You just kind of run it over the whole time. Actually, yeah. 11 is our break, right? Yeah, I don't want to run over, okay. So if you run them over and over and over again, and you score everything, this is the true value. Some are above it, some are below it. So if I have high power, what does that mean? High power means that's my threshold. It means almost every realization is above that. So take the mean of this distribution above that threshold, it pretty much sits on the true value. I don't have an overestimation, or if it's really, really tiny. So when power is high, if you declare a marker to be significant, you're not overestimating its effect. When power is low, what does that mean? Suppose power is 7%. It means only 7% of the time am I going to be above my threshold. Why? Data's noisy, my sample size isn't big enough, lots of things. So when I condition on being above that threshold, this is my new mean. That new mean massively overestimates the true mean. And Shizung Yu and other people have shown how you can directly relate this to the threshold cutoff value under directional selection. And chapter 18, of course, talks about that. So the Beavis effect says you've got to be really careful because your big effect might be an overestimate. Let's go back to Mimulus cardinalis here. So there are the numbers. The original experiment had 93 individuals. They redid the experiment with 465 F2s. This is the line of equal effect. What you'll notice is that the smaller experiment all had larger effect sizes than the larger experiment. In fact, some of them were non-existent larger experiments. This is clearly the Beavis effect in action. You've overestimated your effects by only focusing on those you declare to be significant. So a lot of those major genes people declared were simply statistical overestimation. The second problem they had is fractionation. You've got a QTL, got a nice big peak there. You get all excited. A lot of this stuff came up in looking for speciation genes, where they really put a lot of effort into it. They got all excited. On the X chromosome, I've got this region for a speciation gene. So they find map it. What happens? That one big peak is two smaller peaks. That's okay, I've got two of them. They take one of those smaller peaks, what happens? That smaller peak becomes two smaller peaks. They fractionate away. Your lack of recombination is taking large blocks of effects influencing the trait and having them be co-segregated. If by chance they tend to have a whole bunch of plus alleles, the effect is overestimated. So you get overestimation from Beavis effect, and you're not looking at genes. That L in locus is a misnomer. It should be a location, not a locus. It's not a single site. It's a big chunk of chromosomes. So typically, 10 to 40 centimorgans, and think about that, that's 10 to 40 megabases. So poor resolution, 20 centimorgans or greater was the typical design. Sample sizes in these typically were 300 or smaller because you've got to look at one family. Fine mapping requires making special crosses like nearly isogenic or recombinant inbreds. Line 18 talks about that, or enormous sample sizes. So suppose if the marker QTL are half a centimorgan apart, that means a C of 0.005, C a half of a percent. I need about 3,400 individuals to have a 95% chance of seeing 10 or more recombination events in here. 10 or more recombination events allows me to separate sites that differ by about half a standard deviation. So you can't do fine mapping with QTLs. QTL, that is linkage mapping, gave us a very biased picture about it, basically overestimated the fraction of major genes which were there. So if you read the literature from the mid-90s, you have a very different feel from if you read the literature now where people have used other methods. People got all excited. Quantitative genetics is dead because we can find the 6 or 7 genes underlying this, and Mike and I are out of a job, and so are most of you. But it turns out that that's not the case. Most major QTLs fractionate. QTLs, a large effect, are routinely discovered, but upon further mapping, they break up into multiple fragments. Now, there are big QTLs which are found, certainly true, but as often as not, a major effect QTL fractionates and often fractionates away when you find mapping. Then you have the Beavis effect. If you declare a QTL to be significant, you significantly overestimate it. Now, one implication of these two is that when QTLs were first discovered, a lot of the plant breeding companies went big time into marker-assisted selection. They lost a ton of money because most of the things they tagged were overestimation and they would segregate away. So a lot of the impetus for getting better methods came out of people trying to get money in the agricultural literature. So questions about the drawbacks so far about QTL mapping. It's a cool approach. And by the way, it's not without use. A great use of it is people have shown, for example, that speciation genes are disproportionately found on the X chromosome. That's a real nice use of QTL mapping. Some of these species, the back crosses one direction are viable and not the other way. So you can use these to try to map speciation genes. And likewise, you can basically ask, if you, do you have chromosome regions that tend to show a big effect? Well, those would be areas to look more further studies on. But for most applications, especially in evolution, the sample sizes were small and people got overly excited. They're clearly good, true positives of things with big effects, but there's also enough noise out there that a reviewer can be very suspicious unless you do a lot of hard follow-up work. So that was where QTL mapping sat. Questions or comments on that gloomy perspective? Got to make you, it's like any good sports story. Your team has to do really bad before they come out the end to get really busy. Mike.
Did you want to say anything about the thresholds used here, the number of markers that are used, the number of chromosomes?
Yeah, so we'll talk about the good. So Mike was basically commenting. So how many markers do you use? The bottom line is for QTL mapping, you need, for most chromosomes, just a couple of markers per chromosome. Why? Because you're not going to get more recombinations given the modest sample size. When we go to GWAS, it's totally different. GWAS is in the tens of thousands at a minimum. We'll get to that. Because remember, in QTL mapping, you're looking at one or a couple of generations of recombination. So big chunks get carried along by most individuals. So in QTL mapping, I typically say, 10 markers per chromosome, unless you have unusually long chromosomes, is probably just fine. Because QTL mapping, you're not trying to find map. You're trying to find map, it's not going to work. You need to have a specific reason to do QTL mapping, because nowadays GWAS is a better approach, even in evolutionary settings. Okay. Now, just one more comment. In natural populations, it's often hard to get large families, so you can then do QTL mapping in outbred populations. This requires following pedigrees, and I won't say much about this, but the thing is you have to analyze each parent separately. In a line cross, every parent is a big M, big Q over little m, little q. Every parent is a double heterozygote. Every parent is informative. In an outbred cross, most parents aren't double heterozygotes. No linkage information if you don't have a double heterozygote. Likewise, linkage phase varies. Some parent could be big M, big Q. Some parent could be big M, little q. So in these outbred populations, what you do is you follow the alleles from individual parents, which requires things like SSRs to get more precision. Now, one simple approach people have done is called the general pedigree method. And I set this up now because this is what we use in GWAS to correct for relatives. I have individual I, and I have a chromosome region of interest. I'm going to call that region, the affect that region AI. I'm going to try to compute a variance for that region over my sample. Then I've got the background, everything else. So how do I run this model? This is by Goldgar originally. So what this model says is I look at the covariation between individuals I and J. Well, sigma squared A is the additive variation associated with the side I'm looking at. So now I'm looking at a variance instead of a difference in means. Rij is my estimate from markers of what fraction is identical by descent. So for example, Mike and I in that region could have an Rij of 0.1. Nick and I could have an Rij of 0.5. Lewis and I could have an Rij of 0. I put that in my model. This here is that coefficient of coancestry that Jacqueline talked about. And this is for individuals in the population being related. So I have a two-variance component model. I can fit that by ML. And again, what I do is I take a specific region and I look outside that. I look outside to fit this, so typically other chromosomes to fit that. Or I have information on the pedigree. I fit it for that region. I fit sigma squared A for that region. I ask if it's significant. No. Move on to the next region. So this is a way to scan general pedigrees. And because you're using this sigma squared here, you just need the pairwise relatedness in a general pedigree. You don't need any other information. So this is the approach people use to go from QTL mapping in inbred populations to outbred. It's enormously underpowered. Number one, most individuals aren't informative. Number two, variance takes about 4 times the sample size to estimate with precision as means. So people have done QTL mapping in outbred populations by looking at extended pedigrees, but there are issues with doing that. So first on association mapping, questions? Yeah. Yeah, so polygenic predictions is a little bit better off talking about it in terms of GWAS. But when people used marker-assisted selection back in the old days of QTL mapping, what they would do is they would basically look at a marker value and say this marker, if I'm big M, I expect a +4, big M, +2, little m, 0. And they typically wouldn't add those up over everything. They just look at the major genes. A polygenic score, we're going to do the same approach about marker trait association, but now using GWAS, that is a large random sample from a population, not following a pedigree. And in that, I can then look at specific marker scores. And a polygenic risk score is simply the sum of predicted effects over all those markers. We'll talk about that a lot more probably tomorrow. But that's the connection. So you've heard about personalized medicine. That's what they're trying to do, is get polygenic risk scores. Yeah. Yeah, that's a great comment. And I also want to emphasize too that you're actually looking at two different things in linkage mapping versus the population sample. Because think about loci and cardinalis, those are fixed differences. If you go into loci, you're not going to see any of those genes segregating because selection would knock them down to very low frequencies. You wouldn't see them in a traditional line cross or traditional outbred population. You might see them in a massively large GWAS. So again, besides these issues of oversampling and overestimating effects, you're also looking at different things. The variation you're looking for in the F2 from a line cross is variation that's been filtered by selection. And so alleles of large effect could be fixed, whereas if they had a natural population sample, those large effect alleles would be there, but they'd be really tiny with really small variance effects, and I probably missed them. So it's important to keep in mind you're looking at different things when you try to compare these. Besides all the intrinsic issues, I'm also saying that some of these large effects are also probably true, but those aren't large effects that are segregating at high frequencies. They're large effects that were segregating at low frequencies, most likely, and were picked up by selection. And so when you look at the amount of segregating variation in a population, a lot of your response might seem to be polygenic. What you're really doing is you're taking large effect alleles at low frequencies and driving them to higher frequencies. Maybe not to fixation, but to higher frequencies. And that would give you a polygenic respect, even though the underlying genes have large effects. It's just because they're rare, their contribution variance is really small. And something we'll flush out when we talk about GWAS.
If we push the argument a bit farther, would you be as bold as to say, don't get overexcited over new methods and see how it matches with what we know from previous literature, etc.? If you think of the teachings of this QTL story for future approaches, like when you see a new, exciting, flashy method, perhaps see how consistent it is with what we already know. I have the feeling that the BV study, he did the simulation to prove himself what he knew was the case, that there's not enough power, so we'll detect large effect, and he just did the simulation to prove what he already knew, perhaps, no?
Yeah, so Rebecca Dorje, I mentioned of the permutation thresholds. I think she's now provost at either Carnegie Mellon or RPI, but she had a great line, which is never marry a technology. It will leave you. And that's true. We all get, we all get excited when new methods come, as we should, because new methods give us the opportunity to ask questions we haven't asked before. But you always want to have the right amount of temperament when asking those new tools, because you get, it's something called the something hype cycle about, overexcited and underexcited. I'll dig that up. When you get these new tools, it gives you a chance to ask new and interesting questions. But you also, it's like if someone gives you the key to a brand new sports car, you don't want to drive it really fast. You want to drive it carefully to learn how to drive it. So we've given you the keys, like GWAS is another key we'll give you to these things. And so you want to, new technologies are great and they give us tremendous insight and advance the field, but you also want to be a little bit cautious because it's almost always the case that new technology is radically oversold. People have been excited about personalized medicine for 30 years and it's been really good on some things and hasn't really helped us so much on others. And I think it's probably a good place to stop. And because I'll do GWAS tomorrow instead of trying to rush through a little bit of introduction. Final questions or comments? So raise your hand. How many of you are experimentalists? Don't be bashful. Raise it good and high. How many of you did a power analysis before you did your experiment? How many people got successful results? So Nick had mentioned cryptically about the old back of the envelope. Of course, Nick's envelopes are much, much bigger than what Mike and I have, but because we have limited brain power up there. But you should always do a power analysis. So Appendix 5 in our old book, which is Appendix 5 in the new book, talks about how to do power, and they're actually really easy, because all the things you need to do them with are really easy commands in R, like a non-central chi-square F distribution. You should always do a power, because the bottom line is, I can guarantee your experiment is underpowered. Guaranteed. And then the issue becomes, is it worth doing that experiment? Because oftentimes it is. So what a power analysis would say, suppose I expect that, how big does my sample size have to be to pick out a marker that accounts for 5% or more of the phenotypic variation? It's a lot bigger than you think. But you want to do that before you do it and do an experiment with 50 individuals and realize you didn't find anything, right? You should always do about a power analysis. It will be depressing, it'll be sobering, but it'll be good for you, right? It'll give you some realistic expectations. And it's also great if your advisor comes who says, you know, you should do QTL mapping. You know, I did a power analysis and we're gonna need 10,000 individuals. Can I have the money for that? You know, so it's always, you really should do a power analysis before. Yes. Yeah, that's a great question. And basically it gets also the polygenic risk scores. So there's a new thrill that's gone into animal and plant breeding called genomic selection. If you turn around from estimating breeding values to predicting phenotypes, it's called personalized medicine, same thing, where you basically sum up a bunch of little tiny signals over the genome and get something out of it. We'll talk about why that often works. But genomic selection doesn't work by giving you higher accuracy. It works by speeding up the selection time. So for example, instead of growing up this redwood tree and then scoring how good a parent it's going to be, I can look at its DNA and say, well, with low accuracy, I can predict it's going to be a poor parent, so I can cull it right away and grow a lot more individuals. Most of the application of genomic selection in breeding has been to speed up cycle times or to make selection much more efficient. What you're trading off is lower accuracy using markers to predict something with much improved accuracy by getting basically larger sample size, so you can score stuff much quicker. And we'll talk about that a little bit as well. GWAS and genomic selection, you can basically, the same model, you just flip the model around. Okay, any last parting questions for our tea break? So I'll come back on Wednesday and I'll talk about GWAS and we'll get from that and talk a little about genomic architectures. And then I have a lecture near the end, which is untitled, but it'll basically be, I think, useful tools. So we deal with high dimensional data sets. It'll be things like, you all know about Bonferroni, but people probably know about Sims or Hochberg or other things, or combining p-values. How many people have heard of aggregating Cauchy combining p-values, right? It's a new thing, it's really powerful. So we'll talk about things like that. We'll talk about meta-analysis, talk about model selection, maybe talk about the lasso a little bit. So just give you a bunch of, last lecture will give you kind of a quick introduction to a bunch of useful tools that as you deal with high dimensional data, you might find handy. Okay, great. Tea break. That wasn't the clap, that was just letting you know it's tea break.